Hey everyone, it's Mr. Wolfe here, and in this video I'm going to be going over the scientific practice of modeling with cellular respiration and the scientific practice with constructing explanations with cellular respiration. Before we go into that, let's just look at today's agenda. We got our quotation of the week by Albert Einstein, one of my favorite scientists, which is, "A life not lived for others is not worth living." And then we also have some of the upcoming stuff. Next week is going to be our summative on these targets. You're going to have a little break on Thursday to review, and then Friday is going to be our summative on using models and constructing explanations, which this video is on. So if we look at our agenda, hopefully you've taken the cell respiration content quiz already, and that gives you a gauge of how well you know this content. Remember, you're not going to be able to do using models or constructing explanations if you don't have the content knowledge to help you out with it. So if that's the case, make sure that you make some actions, whether it's doing page 16 if you haven't of your packet, right? Doing the Feynman technique again, going back and looking over the homework, coming in and asking me questions, going to the Learning Center. There's plenty of resources and things that you could do to help prepare for everything. Right? But now let's jump. Oh, and one other thing is that notice that on the cellular respiration quiz is that I added some planning investigation questions on there just to help us out with that practice as well. But let's go on to the practices that we care about in this video today. And before we do that, again, here's just an overview. This is the front page of your packet, and really what it's showing is that, hey, these are the targets that you are going to be assessed on for this unit with cellular respiration, using models, planning investigations, analyzing and interpreting data, and constructing explanations. And in terms of how this unit's going to help you prepare for them, we have a bunch of different types of things, and they're going to be scientific explorations is kind of how I'm organizing them. And right now, we're really just working on scientific exploration A. I put in blue the planning investigations box because we did our investigation on Monday with the Ps, and then you also had a few questions on your cellular respiration content quiz as well. All right. Hopefully this just provides you some context of where we're going. Right, so here's the question that we asked on Monday, which is, what factors affect the rate of aerobic cellular respiration? And we took data on tomorrow and Wednesday. We will look closely at that data. We'll be able to answer the question and, most importantly, explain why. Is there effect between non-germinating peas, light or temperature? And if there is, why? And if there isn't, still, why isn't there an effect? What do we know about cellular respiration that tells us that, hey, there's no way that this would have an effect on cellular respiration? So if you go to page 2 of your packets now, what we're going to do is we're going to go over another example with cellular respiration, and the goal of this is to introduce our targets and then to hopefully prepare you to write a good explanation when it comes to this lab that we started on Monday. All right. So our research question is, what effect does a muscle cell have on cellular respiration versus a red blood cell? So we're comparing how the two cells do cellular respiration. Just to give you a little bit of context, here's an actual microscopic image of a muscle cell. The purple little dots, those are going to be the nuclei, and so these would be actually a few muscle cells strung together. Here's also an artist's rendering of what muscle cells would look like, and then on the right side, this would be a computer rendition of what a red blood cell looks like. And then we've given our data. So I'm going to go to the next slide, and I'm going to skip that really quick for now. And here's again our graphs for our heart cell, and I know it says heart cell. It's supposed to be muscle cell. That's a typo on my part. And then red blood cell. So this is supposed to be muscle cell. And we want to model what's going on in our cells based on the data that we're given. So like I said, is that if you get stuck and you're like, oh, I don't know, well, use your resources. All right. I mean, I would like you guys to have this content knowledge in your head, but I understand at this point you may not, and that's fine. So have your content packet out. If you don't, actually stop the video, go and take out your content packet out and have it right next to your skills packet. So if you look at your content packet, especially page 2, this has like all our information that we need. And notice that if I look at my data, I'm like, okay, so this is talking about oxygen. Where do I see oxygen on my target sheet? I see it right here. And remember, so based on this target sheet, if I don't remember, oxygen's a reactant of cellular respiration. So I could go back here and say, okay, well, if oxygen's going down and it's a reactant, well, that means it's being used up. That means that aerobic cellular respiration is happening, which means I have not only the oxygen going in, but I got glucose going in, and as a result, I'm going to get energy and my other products as well, CO2 and H2O. So that's exactly what I'm going to model here. I'm going to show oxygen going into my mitochondria, glucose going in, CO2 going out, H2O going out, and energy going out as well. And now, not only do I have my reactants and products, I'm showing where this happens. It's happening in my mitochondria. let's just say that we're given some other evidence that says that it's not aerobic, but instead I know that anaerobic respiration is happening. If I go back to my target sheet, anaerobic respiration does not happen in the mitochondria. So if that's the case, I need to show glucose not going in the mitochondria. So if I were to draw this, and if it was anaerobic, I would draw an arrow showing glucose just going into my cytoplasm, right? By the way, this circle right here is going to be your nucleus, just something else we're adding to our cell. Right. But it is aerobic respiration, so I'm going to draw my reactants and products, show that they're happening in the mitochondria. Now let's take a look at the red blood cell. And with the red blood cell, oxygen's staying constant. So if that's the case, if oxygen's staying constant, it's not being used up, cellular respiration's not happening. So in this case, I'm actually just going to put nothing. There's nothing going on in my red blood cell. One thing to actually notice too is that this isn't a typo, is that there's not a mitochondria in the cell. That's the actual case with red blood cells. They don't have mitochondria. So think about why it may not be doing cellular respiration. It really just doesn't have the machinery that allows it to do it. Okay, so we've modeled cellular respiration. This would be an example of what it looks like. Right? You look at the graphs, you try to figure out what's going down, and then show the whole picture, all your reactants and products, and where they happen on your cell. All right, so now let's look at constructing explanations. Our research question is there a difference between how muscle cells and red blood cells do cellular respiration? So I'm going to look at my graphs, and I'm not even going to start with my claim. You might know the claim right off the bat, but if you don't, that's fine. Always start with your evidence. You can't make your claim without really knowing your evidence. So here's my evidence, is that I'm going to see, here's my big pattern I put first, is that oxygen decreases more in heart cells compared to red blood cells, right? We could just see that from the two graphs, but I always have to have components, and this is what a lot of students forgot in our last summative, is you had the pattern, but you just forgot your specifics. So I have to have those, right? In the muscle cells, oxygen decreases from 80 to about 20. This is about millimeters of mercury. Do not worry about what that unit is, but having units, if you're trying to communicate it with someone, is important. You don't need to know what this unit means, but you do have to include the units when you're writing everything. So it decreases from 80 to 20 millimeters of mercury. That's just just a measurement of oxygen over 8 minutes. Right? In a red blood cell, oxygen levels stay relatively around 38 millimeters of mercury in the same amount of time. All right. So there's my evidence, got my pattern and my components. So maybe after looking at this evidence, you're like, oh, okay, here's my claim. A muscle cell does more cellular respiration compared to a red blood cell. Now notice, though, that all you've done at this point is your evidence is oxygen. Okay, so what? So what about oxygen? Remember, you're explaining this to someone that really doesn't know anything about cellular respiration. Imagine you're explaining this to your self 2 months ago, or maybe you're explaining it to a little brother, sister, or cousin who's like in 3rd grade. Right? How would you try to explain this to them? So the first thing is, why is oxygen important? And so I literally said oxygen is important because it is a reactant of cellular respiration. Right? That's why I care about, that's how it relates to cellular respiration. But I still have to go into more detail. I want to talk about these patterns. I want to talk about the decrease, and I want to talk about the fact that everything's constant for my red blood cell. So I said oxygen is important. So I said the decrease in oxygen in heart cells. Sorry, that should say muscle cells. That's a typo on my part. This used to be a heart cell problem. That's why there's still some stuff in here. So the decrease in oxygen in the muscle cells means oxygen is being used up and cellular respiration is happening more because, well, just no oxygen is being used in the red blood cell at all. All right. So I've talked about why my evidence is important. I've talked about how it relates to the claim. Now the last thing we want to do is just maybe explain why. Why would a muscle cell do more cellular respiration compared to a red blood cell? And so, I mean, we could kind of just think about it as that muscle cells, in terms of what they're going to be used for, they're used for getting you around, for exercising intensely, right? They require a ton of energy. So it makes sense that they would be doing more cellular respiration, right? Compared to a red blood cell. So I said something like this, is that the cell respiration happens more in a muscle cell because they need more energy in order for us to move around. Also, by the way, if you look, I gave you a few hints too. If you look at the bottom of page 3, it shows you some information on muscle cells compared to red blood cells. Red blood cells, and I mentioned this before in the video, they actually don't have mitochondria. Their only job is to carry oxygen. So if they don't have mitochondria, they're not going to be doing cellular respiration. So either one of these explanations would be fine for convincing someone why muscle cells do cellular respiration more compared to red blood cells. All right. So that's it for that's our intro to constricting explanations. But really quickly, this is on your next page. So this would be page 5, and this is going to be homework. In this question, your friend is trying to make yogurt, but it's not working well. She needs a culture, which is just a sample of microorganisms that does a specific type of anaerobic respiration, specifically lactic acid respiration. She knows that the culture is doing something, but she thinks it's doing the other type of anaerobic respiration. So she thinks she got like a wrong type of culture that's doing alcoholic respiration. So you bring the culture, you help her out, you bring the culture to Stevenson, and you ask your teacher if you could use the CO2 sensor in chamber, and you put the culture of microorganisms just on a plate, and you grow them, and you measure the amount of CO2 before and after. But you also end up measuring the amount of pH. Remember, so that's just the measure of acidity, and the lower the pH goes, the more acidic your substance is. It means you have some type of acid. Okay, so here's the evidence that we get after growing the culture after 3 days. What I would like you guys to do is 2 things. Draw the model, so show what's happening in the cell. Notice that I didn't draw mitochondria because I just don't know if this organism has one, so I didn't put it in. But you want to draw the reactants and products that are happening once you identify the type of respiration this is. All right. Remember, use your content. Use your resources like this, right, to help you out in terms of what could explain the patterns or the things in my data. So draw your model, and then when you're done drawing your model, actually work on constructing an explanation, just like we did up here. Right, so hope this video was helpful. Give page 5 an honest go, and I will see you all tomorrow.